But what are you going to do with it?
Just kind of look at my own score.
You don't have to go to such a big struggle to look for one number. I can tell you.
Well, I did really bad. I don't want to see it and remember it. No.
That's good. 19 out of 23. Out of 23. Perfect. Huh? You did well.
No.
19. Okay. All right, let's start. Ladies and gentlemen, let's start. Okay. So we did replication, we did transcription, and we did translation. Replication is by itself. Transcription and translation are related. They're for one purpose, to make proteins. Okay. Now let me ask some questions. What if there is a mutation? Mutation means mistake. What if there's a mutation in the promoter sequence? What is the result of that? What's the normal sequence of promoter? I think so. Let's see. Yeah, stop. So if it stopped, that's good, right?
Yeah.
Stop codon, not yet. Okay. So let's say you make a mutation. Okay. What's the end result of this? So this is not a promoter sequence anymore. What happens to polymerase? They cannot attach. Can transcription occur? No. So the end result is there's no product, whatever the product might be. If it's lactose, no lactose. You got it. But you have to speak up so I can hear you too. Okay, that's one thing. Now, promoter can change such that, the affinity of polymerase decreases for that sequence. Or it could be mutated that the affinity increases. Okay, so if affinity increases, that means there will probably be more transcription. Affinity decreases, less transcription. Okay, so that's one thing, that's promoter. How about if the operator is mutated? What's the end result? So look, if promoter mutation, let me write it down. So maybe I should make a chart, right? That would be a good idea. So we're talking about mutations in promoter. So let's say mutations. And then result. What is the end result? How about if there's mutation in promoter? What happens to transcription? So decrease mRNA. Correct? Decrease transcription. And of course, this is immediate. And if there's no mRNA, that means there's no or less protein synthesis. That's specific protein. Most promoter mutations are usually very specific, and that means the sequence change, the RNA polymerase affinity decreases. It's extremely rare. Yeah. So promoter mutation will usually lead to what? Decrease transcription, decrease translation, decrease protein product, decrease mRNA. But of course, if you decrease mRNA, the subsequent steps will automatically decrease, right? So we don't even have to discuss that. If there is no transcription, how could you have translation for that specific gene? The prerequisite for translation is what? mRNA. If you don't have mRNA, you cannot have protein synthesis. You cannot make protein out of nothing or from no code. If there's no code, no instruction. Okay, so that's promoter. How about the regulator? If there's a mutation in the regulator sequence. Defective repressor. Very good. So that means repressor activity is decreased, right? Or repressor concentration number is decreased, or repressor that is the same but it's defective. When I say activity, meaning the number of repressor is the same, you can produce repressor no problem, but the repressor is inactive, it's defective. There's other mutation, so you can have mutation where the activity of something is decreased, and you can have other mutation when you cannot produce a product at all, or decrease product. Does it make sense? Yeah. So regulator mutation will lead to decrease in repressor. What will happen to mRNA? Very good. So the end result is increased transcription, increase in mRNA, and increase in protein synthesis or product. Right? Okay. How about if you have mutation in the operator? The operator sequence is changed. Repressor can bind. So what happens to transcription? Increase. And protein synthesis will increase as well. And when I say protein synthesis, that does not mean all proteins, that specific protein. Okay? Because each gene has its own operator, its own promoter, its own regulator. One promoter is not dependent on the other promoter. Promoter for insulin has nothing to do with promoter for glucagon. They're independent. Okay? Okay. What if there's a mutation such that a repressor is different? Repressor, which is a protein, right? Repressor is defective. Now you can have two types of mutation because of repressor. One is gain of function, one is loss of function. Let's say, number one, a repressor is mutated such that it can no longer bind to the operator. That means, so let's say in this case, the operating sequence or the operator binding sequence is changed, right? So the repressor is mutated that cannot bind. Can't bind. Can't bind. That means loss of function. Can't bind its job. What is the job of a repressor? What is the function of a repressor? To bind the operator and prevent transcription, right? So if it's loss of function, that means it can't bind the operator. Its normal function is what? Bind the operator. If it cannot bind to the operator, that means it loses its function. So loss of function mutation for a repressor will lead to increase in what? Transcription. Increase in protein synthesis. So that's one. Now what if a repressor so one mutation is that the repressor can't bind operator. The other one is repressor can't bind inducer, which is what? Gain of function or loss of function. Yeah. Okay. So in this case, repressor cannot bind the inducer. If you cannot bind the inducer, that means repressor is normal. It can bind the operator, but inducer cannot deattach it. Okay, the repressor function increased, but still there is decrease in mRNA. Right? So this is considered loss of function. Loss of function, yeah. Okay, so it's simply, if the mutation is within the repressor such that it cannot bind to the operator, it's something different than when the repressor is mutated such that it cannot bind the inducer. The results are different, but it's mutation regardless. If it cannot bind the inducer, that means inducer cannot attach to it. Repressor will stay attached, bound to operator. Okay, so again, let me just here you go. This is mRNA, and this is the region of operator. And you know that a repressor binds where? To the operator, right? It binds to the operator and it blocks the operator. Okay. One mutation is such that the repressor cannot bind the operator. If it cannot bind the operator, that means this repressor is free. It cannot bind the operator. That means transcription will continue because you cannot block polymerase. You cannot block polymerase. So that's one option. The other option is the repressor cannot bind inducer. It could bind the operator, no problem, but it cannot bind inducer. And inducer is supposed to inactivate repressor. So if inducer cannot bind to it, that means there will be no transcription because operator is occupied and transcription is blocked. So repressor have two binding sites. One is inducer binding sites and one is operator binding sites. So it depends which sequence is changed. Good? Okay. And you see it's not difficult. It's just because there's a lot of information in terms of it makes people confused and scared. But at the end of the day, it's the same common sense. You know? Okay. So that's simply with the initiation of transcription. These issues can occur. Now, if you have, for example, mutation in promoter sequence of growth factors, that means growth will decrease. If you have mutation in the regulator or operator of growth factors, growth genes, you will have too much growth. That can lead to cancers. So usually when you have, for example, mutation in the operator, and that leads to unstopped, uncontrolled growth, it can lead to cancers. So some cancers can have operator mutations. Okay. At the end of the day, if you have too much protein, and those proteins are responsible for growth, that means cancer. If the proteins are responsible for digestion, then we increase digestion. I'm only talking about operator sequence change or mutations. Okay. So you can apply. Basic is what? No operator, no regulator, no promoter, no repressor. If you know them, it doesn't matter how I ask it. Basics are basics, right? Once you know the basics, it doesn't really matter. You should be able to apply it in any situation, any circumstances. But I'll give you questions for practice. Okay. Okay. Yeah, people sometimes usually, a lot of people hate biochemistry because there's so many small details. It just simply, it's like on and on switch. If you have 3 switches on, 1 off, what is the end result? Still on. That's, if you have 20 off and 19 on, what is the result? Still off. Or partially off. You know, that's where it comes to. How well you understand the concept. So that's one thing. Let's go now to what if you have mutation? Oh, I should have kept that last list. Let's see. What if you have a mutation in polymerase, RNA polymerase? So RNA polymerase also has a promoter sequence or promoter binding area. So if that region is mutated, that means polymerase cannot attach to promoter. You have the promoter, it's perfectly fine, but polymerase cannot attach to promoter. Again, no transcription, right? Okay. Or let's say you take medications that inhibits, covers up polymerase binding sites or promoter binding sites on polymerase. Let's say this is a polymerase, and right here, this region, red, is the region that binds. That's not red. This area is the region that binds to the promoter. And then you take medication known as rifampin, for example, which is used for TB infections and stuff like that. And this thing comes in and simply covers this area. Now this polymerase is ineffective, can't do anything. But it doesn't have, see, rifampin doesn't bind human polymerase, it only binds bacterial. So it will only kill the bacteria, or at least prevent bacteria from growth, wouldn't affect your cells. And anytime when we talk about things like that, if you use agents that prevents transcription or inhibits transcription, it will only act on those cells that are transcriptionally active. If a cell is not involved in making protein synthesis, then it doesn't matter. This medication will be ineffective. It would only act, or this agent, it will only act on those cells that are transcriptionally active. cells that make more protein. Make sense? So you have to understand all circumstances. You don't find cells in the typical cases. Your body is never typical. It varies on situations, sometimes this, sometimes that, sometimes this. But the end result is you got to know these basics. Application is easy once you know the basics. You have to know the basics. Okay? And something about rifampin, if you can remember it for later on, everything is with R. It turns you saliva, red color. Tears, red color. Urine, red color. Usually patient, when they're suspected of TB and you give them this medication, they will come the next day or a week later to clinicians or your clinic or your hospital. I have bloody urine. And then people think either they have kidney stones or they have cancer, they get scared, or infection, but it's nothing like that. It's simply medication. Stop it and it will come back to return. Rifampin, red. So think of R for rifampin, RNA polymerase inhibitor, only in bacteria. R for red urine, red saliva, red tears. And on Halloween days, this is a great medication if you want to scare people. Cry red. You can use it. You know? So, rifampin, RIF, and then AMIPIN.
But it's not over the counter.
Okay, so if you have a doctor friend and you want to go on a Halloween thing.
Yeah, you can cry red, be red.
Yeah. Yeah. Spit, red, all of that, yeah. So please, now you see there's a lot of information, but it's how you use it. So mutation promoter you should know, mutation regulator you should know, mutation in operator, repressor, and polymerase. You should know this mutation. Only if you know their normal function, right? Just give me a second. Only if you know their normal function, mutation doesn't matter. If your job is, as an athlete, where's Ahmed Farah? If you want to run 100 meters in 20 minutes, like you do usually,
100 meters in 20 minutes?
that's what he does.
100 meters, 20 minutes?
Yeah.
Sure.
Total.
Okay, that's what, okay. But he does it in 20 seconds. Yeah.
But it's bad.
It's bad still. Okay.
20 seconds? Yeah.
Should be 5 seconds, 6 seconds. Anyways.
Average is 13, yeah.
Yeah.
No,
Good.
13 for
Anyhow, if your job, look, if I lost my thought now. I don't know what I was saying.
Yeah, if you know the normal function.
Yeah, if you know the normal function of something, as soon as you have, for example, let's say, you know, your job is, you're a surgeon. You're a surgeon, and you get your hands traumatized in a motor vehicle accident, then the job of your hands is to perform surgery. You won't be able to do that. You can do other things, but you can't do that. So always focus on normal function. Always focus on physiology. Once you know physiology, the abnormality is not a problem. Path is super easy and it's interesting. If you don't know the normal function, you will suffer so much. Okay, so focus on what is the purpose of promoter, what is the purpose of regulator, operator, repressor, and polymerase. Okay, transcription factor, for example. Transcription factor, I mentioned many, many times. They help polymerase attach to promoter. It doesn't mean they cannot bind anymore, but the binding is not strong enough. It's weak. So it will affect transcription. If not completely, it will partially affect transcription. Okay? So now, in addition, I don't even have to tell you this, but sometimes it's a good reminder because I assume you can think that if you have polymerase, they should have promoter binding area. They should have transcription factor binding area. They should have these automatically. Otherwise, nothing would happen. So even without me telling you, you should be able to reach those conclusions. But again, if you study under stress and you think you've done your job, that's not good enough. Once you study, you have to think, how is this information related? You really have to give yourself time to think and apply and conceptualize and think about it, especially visualize. You know, there's a lot of videos that should be available on Google, on YouTube, so whatever that is, regarding these things. Watch them. I think that will really, really be helpful. It should be really helpful. Because when you see with your eyes, is the best evidence is your vision, right? And it relates. And you learn more better too. Okay. there's more neuron coming from the visual cortex to the memory cortex than anywhere else. Not sound, not feeling, it's not smelling, it's vision. You learn much, much more with vision. Secondary is hearing. But the hearing cortex is more stimulated by your own sound. Not by my audio, by your audio. So if you hear your own sound loud enough, it's unbelievable to your brain, because your brain loves you. It doesn't love me, it doesn't care for me. It doesn't care for anybody else, it cares for you, except your brain. So you have to listen to yourself. Repeat it louder as much as you can. It will help you, trust me. Just try it. If you haven't tried it, try it. And those of you who have families and if they get bothered, just get insulator or something, I don't know. Okay, what else? So know these things. Know these mutations. Let's move on to now the transcription itself. So this was before gene. Now if there is a mutation in the gene itself, look, mutation in promoter, you don't even get an mRNA. Mutation in promoter, you don't even get mRNA or you get too much. All the mutation that I talked about right now, you get mRNA or you don't get mRNA. Now, so that means you get protein or you don't get protein. What if there's a mutation within the gene? The sequence of the gene, let's say, is A, C, C, U, G, G. Since we're talking about mRNA, so I'm using U. So G, A, whatever it might be. U, A. Okay. Now, there's multiple things that could happen. Let's say there's a substitution mutation in the second codon, okay? And they will tell you second codon. There's a mutation right here. C is changed. Instead of C, you have, let's say, G. The amino acid that's coded by this is different now. The product will be different. Okay, the product will be different. Let me give you an example. This is for sickle cell anemia, for cystic fibrosis, for example. Okay, for these are called substitution mutation. What else can I think of? Herb Duchenne, and then you also have what's the disease of muscle when you have big calf muscles? Duchenne muscular dystrophy. So you can have these single nucleotide mutations, and the product is different. Okay, because the amino acid sequence is now changed. You suddenly have an amino acid that shouldn't be there before. Instead of having a normal amino acid, you have abnormal. This amino acid doesn't belong here, so the folding is changed overall. Okay, so that's one thing. You can have one mutation. Yeah.
If you say this is a gene, the mutation's on the gene, shouldn't this be a DNA?
Well, gene is also in the RNA. Yeah.
Well,
You have it in DNA, but it also transcribe into mRNA. Yeah. Of course, this should occur where? In the transcription process. So you can have, for example, in the transcription process, let's say the RNA polymerase substitutes, instead of C, a G. That's a problem. For mRNA, there's less proofreading. For DNA, there's more proofreading. And it makes sense too, because mRNA is not permanent. You don't need much proofreading for that. If you make one mistake, it's not a big deal. It won't kill you. You won't inherit it and you won't give it to your children. But if the mutation is in the DNA, it's a lot more problematic because it's permanent. So you have more proofreading, more regulation in DNA synthesis than RNA synthesis. So the likelihood of mutation in the RNA is much, much higher than in DNA. Likelihood of mutation or substitution. So look, mutation can be called substitution if you substitute one nucleotide with another one, correct? And if you remove the entire nucleotide, what would you call it? Deletion. You can just move the whole thing, deletion. By mistake, well, let's say you add one extra, what would you call that? Addition or insertion. These are all good words and it makes sense. You can have addition mutation, deletion mutation, or substitution. Now, these mutation, when it's a single nucleotide long, when there's a single nucleotide change, whether it's insertion, whether it's deletion, whether it is addition, they're called point mutation because it's a single nucleotide change. Point mutation. Point mutations. Okay? Point, 1 point. One nucleotide. Let's say a sequence. I'll make a space. A, U, G. So think of DNA the same way. So let's say this is DNA, or this doesn't matter. And then let's say have Q. So let's say we have a point mutation. If you have point mutation can be substitution. Let's say instead of, now I'll make him in red, the bad guys. There you go. Instead of C, you have G. What happened is, that means if it's the mutation is the first two nucleotide, you got missense mutation. Missense mutation. Remember that the amino acid specificity on codon is based on the what? The first two nucleotides. If you change the first two nucleotides, the likelihood of changing amino acid is very high. The first two nucleotide of a codon is specific to each amino acid. The first two nucleotide determines the amino acid. So if I make a mutation in the first two nucleotide of a codon, obviously, let's say CC was for cysteine, right? CC was for cysteine. Now you have GCC. Do you guys have the code? Let me just open your book. I don't know which page you have the code, and let me tell you how you read it. So that way we can do this as a practice. Otherwise. Page 90, page 90, page 90. This is the hard part, application of it, okay? But you have to know well so you can apply. You see on that page, you see in the page 90, there's a chart. On page 90, there's a chart, okay? And you have this genetic code. You know what, I think I should put it on my screen. Should I? And it says vertically on the left side, it says first codon. First codon. Let me see if I can find it. Let me see if I can find it. 90. 90.
There are two 90s, so it's the first one.
There's two 90s?
Yes. It's because the GI system stands for.
Oh. 90. Okay, so this is the chart I'm talking about. You see the first, tells you first nucleotide. If here we have, what do we have before? CCC, right? I can't erase anymore because I pasted. Wow. The first nucleotide was CCC, right? Let's say, now the first nucleotide is C. Let's go to C right here. C. So we'll start here with C. So the first nucleotide should be C. Here you go, that's C. What's the second one? C. So that means in this box, right? C and C right here. And what's the third one? C. And the third one, C, is, when you look here, is right here. Okay, so this one is coding for what? CCC? So, very good. Very good, proline. Okay, so that's what we do. Now, I went too far on my thing. So there's first C, and you come here. These are all C's, and then all C's here, and then third C's right here. So you come, everything matches here, proline. So it was proline, and when I change the first nucleotide to G, look, it becomes valine, or alanine. Alanine, all four of them, doesn't matter. So we switch from from proline to alanine, just by substitution of a single nucleotide.
Can you do an example for Huntington's since you have that chart, please? Because if it's the CAG trinucleotide repeat, how would that work?
Okay. Now in Huntington, it's not necessarily it's not a point mutation. Okay? In Huntington's, you have trinucleotide repeat. These trinucleotide repeats are over and over and over and over, and it destabilizes the gene. It's nothing to do with the sequence. It's just you have too much repetition of something that shouldn't be there. Okay? And it makes the gene unstabilized, so you have non-coding regions. And the product is unstable. So, for example, if it has, I think it's
See, isn't
CAGE.
Isn't
It's CAGE, yeah.
glutamine or
Yeah, this stands for glutamine. So you have too much glutamine and the product is too soluble. That's all. Okay. All right. Now, so that's one. Look, substitution will lead to what? Missense mutation, if it's in the first two nucleotide. But if I change the last one, it wouldn't make a difference. Especially if it was, let's say if I change the last C to U. If I change the last C to U, it doesn't make any difference. The product is still proline, and that is silent mutation. So mutation in the third nucleotide of codon usually would not change things. Okay? Especially if that amino acid has more than one codon. Okay? Tryptophan is the only one that will be problematic because it has one codon. Other ones have more than one codon, so that's one thing. Now, that was substitution, but guess what? What if you do addition or deletion? Yeah.
I have a question. So you said that on the last one, if it gets changed to silent mutation, but what if, like, for example, if we look at CA, then C, it's histine, but then if it changes to CAA, it's, glutamine.
Yeah.
That's still silent.
That's what I'm
mutation?
saying. No. As soon as you change the first two, one of the first two, amino acid sequence changes.
It's not changing one of the first two. It's the last one. Isn't it the last one that's changing?
Okay, so if you change, remember we had CCC, right?
Okay.
So if you keep C the same and you change this C to A, and it's the same. This one was CCC is for what? Proline, right? It codes for proline. CAC codes for what? It's a different amino acid.
No, I'm saying, like, if, for example, if you put CCC,
Mhm.
right, and then on the second one you put CCA.
Okay.
If they change
Yeah.
third,
CCA, right?
Yes.
So this was proline, and let's say CCA. CCA will be arginine. CC, CC, A.
I meant,
Oh, it's also,
CA.
It's gonna be the same.
CAA.
Yeah, it's still A.
Yeah, but it
yeah, so proline doesn't really, in proline none of them will change. Because proline has 4.
I apologize, I meant CAC and then CAA.
CAC, and then CAA, right?
Same.
Yes.
Okay, so look, as soon as you change the second nucleotide, first of all, you just want to talk about between these two, right?
Yes.
Okay, if you want to compare these two, let's see.
It shouldn't change.
What is CAC?
Okay,
CAC. Histidine, good. And what is CAA? Glutamine. So in this case, because histidine has only 2, proline had 4 codon, yeah. That's what I'm saying. So it depends. In some cases, the third nucleotide might not do anything. In some cases, it will. But as soon as you change the first 2, no matter what it is, it will change the amino acid. Yeah? Okay. But these are called point mutations. Substitution is not a big deal. It wouldn't change much. The problem is, what if you do deletion or addition? Can't even erase this. So let's say we add here A. It changed the whole frame. Very good. The reason for that is because now we have to make 3 codons at a time. If you add A, now this is different. 3 at a time. We get a completely different frame. And these are devastating mutations. Okay, so Duchenne muscular dystrophy is usually that. Marfan is because of that. So there's a lot of disease that can occur because of the whole product is now different. They're devastating. So that's similarly, that was if you add a single nucleotide. What if you delete a single nucleotide? Same thing.
that's it.
As long as addition or deletion is not 3 or it's multiple. Because if you add 3, you didn't change the whole frame, you just added one single amino acid somewhere in there. You didn't change the whole sequence, right? Just one amino acid is different. One additional amino acid. If it's 3 or it's multiple, then it leads to, it's okay. But if it's not 3 or multiple, it's 1 or 2, it changes the whole frame. Make sense? Yeah. Okay, one more thing. What if you have premature stop codon? Let's say instead of, this is this is A. Instead of that, instead of A, you change this to UAA. Now the stop codon should occur somewhere here later on, but it's a premature stop codon. This will lead to a small protein, maybe 1/4 of a protein, half a protein, and they're not functional. Nonsense mutation, yeah. Okay. Silent mutation, the protein product or the amino acid didn't change. Silent mutation, amino acid sequence did not change at all. There was a mutation, but amino acid did not change. It's silent. Missense, it still makes sense, but it's missense. Instead of being alanine, it's glutamine. Silent, amino acid sequence did not change. Maybe mRNA changed, maybe DNA changed, but amino acid sequence did not change. That's silent. Missense, you misinterpreted the amino acid. Instead of saying glutamine, you have valine, let's say. So amino acid sequence changed. Nonsense. It makes no sense whatsoever. Stop codon. So stop codon is called nonsense mutation. Again, not normal stop codon, premature stop codon. Premature. Okay? Let me give you examples in real life situations. So let's see if I can clear all this. Whoa. Clear, right? It doesn't do it. Maybe new. I think in cystic fibrosis, we can maybe do example of cystic fibrosis, and let's do example of sickle cell anemia. Okay, so these are the one I should probably remember. In sickle cell anemia, it's clear cut. Glutamate is substituted with valine. Instead of having glutamine or glutamate, you have valine now. Okay, which means this is a point mutation. Cannot be frame. Frame shift will change the entire product. And this must have been in the first 2 nucleotides. Okay, depending how many codons. Let's see if I still have that thing. Paste. Do I have it? No. Oh, I have it. So, where is glutamate? Glutamate. There you go, glutamate. And then it changed to valine. Yeah, so if it's if it's U, it'll be valine. As soon as you change U to, for example, to either A or you change it to A, yeah, if you change U to A, it becomes glutamate. Okay? But it's not in the last nucleotide. It's usually have to be in the first one. And this leads to an insoluble protein. It makes a lot of difficulties, a lot of problems. And this goes for hemoglobin. So hemoglobin is supposed to carry oxygen. It's supposed to carry oxygen. Now this hemoglobin that has valine is more lipid soluble compared to glutamate, which is water soluble. The amino acid that has, the hemoglobin that has glutamate is more soluble in water, has no problem, can swim, can carry oxygen. But the one that has valine now is lipid soluble. And then if this is a cell, just let's think about it. If this is a cell, and here you have the hemoglobin, because the cytoplasm, I'll make it blue. The cytoplasm of RBC is what? Water, right? Because it has no cytoplasm. It's pretty much all cytosol, actually. The cytosol is only water. And I'll make one hemoglobin. This H is normal. I'll put HN, normal. And then, you know what's Yeah, let's just use that. So you will have hemoglobin adult, let's use A, and for disease, hemoglobin is S, which is sickle. That's the term they use for, but anyhow. So S is defective, A is normal. Okay? Now you know that blue is normal, it's soluble in water, so it has no problem swimming around. Red is lipid soluble, it's defective. What is the membrane made of? Lipid, so it would attract this. Upon deoxygenation, the S goes and it fuses with the membrane. And if a lot of them fuses, it allows the membrane to become stretched and sharp. And instead of having round cells, they become crescent shaped. It's simply because of that, simply because of solubility. And technically, all things that we call toxic in our body are things that are insoluble in plasma or in fluid. For example, when you have bilirubin, if it's unconjugated, it's insoluble. It can cross the blood-brain barrier and deposit there and cause mental retardation in children. Iron, if it's too much, it's insoluble in some cases. Combining with some stuff causes problem. That's where toxicity comes from. Although, unless you activate the receptors and stuff, everything is okay in small concentration. Yeah.
How does mercury and lead affect this? That's similar to mercury and lead as well.
Yeah, mercury and lead actually, number one, they can cause membrane peroxidation, lipid peroxidation, which causes damage to membranes, a lot of membranes, called cell lysis. But it has special predilection for myelin sheaths or brain tissue. Okay, so in GI, it will cause different something than the brain. Yeah. But enzymes and proteins requires divalent metals, magnesium, copper, iron, lead, or mercury. So mercury is not normal, lead is not normal. Most enzymes in your body would use magnesium, iron, or copper. That's good. What happens is mercury and lead have similar structure to those things. When they bind to the side of iron binding sites, magnesium binding sites, things like that, it will inhibit the enzyme from activity. Okay? In sickle cell, the damage is in chloride channels. Okay, so chloride channels. And you have cells called exocrine cells. Okay, this is not on the exam right now, just for you to understand the idea how mutations work. We have sweat glands, right? Okay, so let's say this is a sweat gland. You have tear glands, and you have glands called mucus glands, or mucus cells. On their surface, they have chloride channel. Yeah, cystic fibrosis. No, cystic fibrosis, my bad. If I said sickle cell, I'm wrong. So this is supposed to secrete mucus. Mucus. Okay, it's supposed to secrete mucus. When it secretes mucus, then, for example, and you're supposed to also secrete chloride in there. So when you have chloride, water will follow it, and the mucus will stay thin. As long as you have chloride in mucus, water follows it. Remember, water always follows sodium chloride. Everywhere sodium chloride goes, water follows it. So if you have enough chloride and sodium in here, especially chloride because these are chloride channels, then that means the mucus will stay dilute. What if the chloride channel is defective? That means you have mucus, you can secrete mucus, but chloride is not there, so mucus becomes thick. It's a thick mucus. So if this is saliva, saliva will be thick. If this is sweat, thick. In sweat, actually, it's different. You cannot take chloride, so it stays very thick. Water is absorbed, but chloride is left alone. So it will taste very salty. Okay? So again, it stays thick because there's no water that can follow because chloride. And then in GI and in respiratory system, let's say this is respiratory system, in GI, again, mucus will stay thick. See, the problem is it's not a big deal whether it has chloride or not. The problem is thick mucus. Thick mucus. And this mucus will block tiny little pathways or channels or tubes. In GI, it can cause constipation because it will block passage of stool. When you have thick mucus, it's different than thin mucus. Thin mucus can flow easily. It's almost like having, taking shower with pure water or taking shower with jelly in your pipes. When you turn it on, no jelly is going to come out. It will block water flow. Okay. So the end result is it's not because of the chloride channel that we need chloride, it is the thick mucus. So the consequences of that, and this will cause respiratory infection, constipation, and so forth, and it will block pancreatic ducts, will cause malabsorption, and so forth. But let's talk about how is this related to protein synthesis. Okay, how is this related to protein synthesis? So first, you do the transcription, the product is normal. During the transcription, something can happen. Let's say splicing doesn't occur properly. If splicing doesn't occur properly, that means you have non-coding regions in there. And the product that you'll make is the cystic fibrosis transregulator protein or chloride channel. I'll just use chloride channel. Chloride channel. This chloride channel will now fold differently. If it has the appropriate sequence, the channel will make appropriate channel. Nice, clear-cut channel like this so things can go through it properly because it folded properly. But if you change the sequence, then it will appear something like this. It's no longer a channel. It's misfolded protein. So as a result, you cannot transport or excrete chloride, and that leads to the devastating effect. But the mutation can occur. Again, it could be a single nucleotide mutation. It could be folding problem, or it could be splicing problem, or it could be promoter problem. But most of them are folding. Okay, most of them are folding. And guess what? There's additional information you guys did, cell membranes. So you're supposed to put this information together. We did cell membranes, right? We talked about cell membranes. Did we discuss transmembrane proteins? At least we talked about G protein, right? A G protein is supposed to have extracellular domain, I'll put E. Intracellular domain, I'll put I. And then transmembrane domain. The transmembrane domain is supposed to be made of what?
Phospholipid bilayer gets neutral amino acids.
Neutral amino acid. So now, when you substitute in cystic fibrosis, because it's a channel, it has to be a transmembrane protein. It's a chloride channel. If the domain here is defective, this protein will not even stay in the membrane. So the protein is made in the ribosomes, and then it's transported from Golgi perfectly into the membrane. When it reaches the membrane, you cannot embed it in the membrane because it has no lipid-soluble amino acids. Then it's returned back to the company, say, hey, fix it. And start accumulating and cause more problems. You just have to take one step at a time. Okay, so there's a lot to it, but depends where the defects will occur. Okay, so in other cases, you heard of Marfan's disease? You heard of, let's say, scurvy? There was a question on it, right?
Yeah.
I'm glad I remember that. So scurvy. What is the issue in scurvy? Vitamin C deficiency, right? Okay. Now, the protein synthesis that requires vitamin C is known as collagen. Collagen. So mRNA is perfectly fine. DNA is fine. The product protein that is made, the linear chain of amino acid, is perfectly fine. You don't require vitamin C for that. Once you make that nascent chain, the primary chain, then it has to fold and it has to be modified, which is post-translation modification. One of those steps in the rough endoplasmic reticulum is when you have this chain, when let's say it folds, or before it folds, it's just alpha helix, and that's the truth of collagen, it's alpha helix, okay? There are OH that needs to be added. This is post-translation modification. The OHs needs to be added, and this is known as hydroxylation. Addition of OHs requires vitamin C. So there was no problem in transcription. There was no problem in protein synthesis. The problem is now what? Post-translation modification. So vitamin C is required for hydroxylation of collagen fibril or alpha helix. Now, you might say, who cares for that? I can't think of this will be a problem because I cannot relate it to real life. Let me tell you. If you have a spring, you have 3 springs. So at the end of the day, for collagen to be stronger, to withstand this sheer pressure of stretching and so forth. Your blood vessels are made of collagen, your muscles made of collagen, your ligaments are made of collagen. They stretch all the time, but how come they don't tear? That means there must be some sort of flexibility. First of all, it's like a spring. You can stretch springs, right? That's why it's coil, alpha helix. Good? Now, if you have one spring and you stretch it, it's stronger. You have three of them put together. Three. So first you'll make the alpha helix, okay? And then three alpha helix have to be combined. Let me use different colors. This is called pro-collagen. Before mature collagen, you have triplets. When you triplet, it's almost like making braids. It's much stronger than a single hair, right? So now, this is the final product. But guess what? If you have a single alpha helix that is simple, straight like this, that is easier to attach or stick together, or when it has OHs. When it has OHs, it's almost like hooks. You can't pull them apart. They stick together. Further, there's glycosylation. Further, there's disulfide bond formation. So at the end of the day, the whole idea is that triplets have to glue together. If you have a linear molecule, like for example these pens, I have two pens, there you go. They're linear, they don't stick. But if it was coiled and it had branches like trees, you put them together, they'll stick together. That's the whole idea. Okay. Okay. Study a little bit at home. If you have questions, it's easier for me, when you have specific questions, to kind of tackle those and give you examples. If you don't have questions on today's what? Today's Friday. If you don't have questions on Monday, then I'll do inheritance. How do you inherit conditions?
Is there going to be exam Tuesday?
If you don't finish genetics by Tuesday, there's no exam. There might be just a small quiz, just to keep you in on thing. Which I don't think, because we still have to finish genetic diseases like Turner's, Down syndrome, Edwards, Klinefelter's, Angelman, Devilman, all of those. Yeah.
All members.
Mm.